Welcome back to sqadia.com. My name is Muhammad Osama, and today we are talking about inflammation. The next topic that we are going to cover in this series of lecture is the cellular response to leukocytes and the hereditary defects which can impair the normal process of acute inflammation. So these are the topics: cellular responses to leukocytes, mediators of inflammation, and hereditary defects, which we are going to cover in this lecture. So what are the cellular responses to leukocytes? There is emigration, chemotaxis, phagocytosis, and intracellular microbial killing. So let's take a depth view of these terms. So first of all, we have emigration. If you remember the previous slide, we discussed that during the process of inflammation, the different type of cells and leukocytes, they come to the site of injury. But how does that happen? That is known as the process of immigration. So what happens is that, first of all, they are attracted by different type of products which are released during the process of inflammation, which are known as cytokines or chemokines. These cytokines and chemokines attract those leukocytes or WBCs. So those leukocytes come to that area through the blood flow, and once they reach there, that process is known as migration, the process of coming to that area or the site of injury or inflammation. Once they have reached there, they are still in the blood vessel and not the tissue. So what happens is that the second process is known as pavementing. In pavementing, they start to accumulate at the site where they want to dislodge from the blood vessel and go into the interstitial spaces. So that is the part which is known as pavementing. And when they start to accumulate in that area, what happens is that different type of activities start to occur in the cell, which is actually known as rolling. Rolling is actually mediated by endothelial cell selectins, which are different type of adhesive molecules as we studied previously. So those endothelial selectins or adhesive molecules, they loosely bind the leukocytes with the endothelial cells, and thus that is why they result in rolling. So they move on in a rolling manner, and that rolling suddenly or eventually stops, and that part is known as arrest or adhesion. During the adhesion or arrest, the cell stops rolling and adheres to the endothelial lining, and that is mediated by another type of adhesion molecules, which are the integrins on the leukocytes to the immunoglobulin family adhesion proteins, which are present on the endothelial cells. So now during the arrest or adhesion, now the cell has become stationary. Then the last part of immigration occurs, which is known as migration or transmigration. It is mediated by platelet endothelial cell adhesion molecules, which results in the movement of this cell from, or a leukocyte, from the lumen of the blood vessels into the tissue where the inflammatory process is occurring or where the injury has just occurred. So this is known as immigration. Now, second is chemotaxis. Chemotaxis is the process by which leukocytes are attracted to one of the injured sites, and so that they can go there and perform their function, as you can see here. So here were different type of cytokines and chemokines, so they were attracting those WBCs or leukocytes. That's how they came to know where I have to go. So this process is actually known as chemotaxis. Chemotactic factors for leukocytes include the products that are actually produced by the bacteria or the complement systems, especially C5a. Other include arachidonic acid metabolites, for example, leukotriene B4. So these are all different types of chemokines or cytokines that actually attract the leukocytes to the site of injury or inflammation. Now, This leads to the function of the leukocytes, which is the phagocytosis. So neutrophils, which are the first respondents in the 24 hours, and monocytes, which come later on after 2 to 3 days in the process of inflammation. So these two type of cells are the most important phagocytic cells. So what is phagocytosis? Phagocytosis is actually the engulfing of that harmful microorganism or harmful protein or any harmful product which is being placed there or produced there at the site of inflammation, so that it can be engulfed and destroyed. Opsonization is a phenomena which actually facilitates this process of phagocytosis. What happens is that there is a coating of opsins which immobilizes the particles on the surface of the phagocyte. So once the particle or microorganism is immobilized and it becomes stationary, it becomes easier for the macrophage or the neutrophil or the monocyte to engulf that organism or that product. Most important opsins that are present are the immunoglobulin G or IgG subtypes and C3b, which is a part of the complement component. What are the anatomical changes that occur during phagocytosis? So during phagocytosis, there is an internalization, which means engulfing of the attached opsonized particle. As you can see here, for example, it is a bacteria. So these are the opsins which have made it immovable or stationary. So it is being internalized. So this here you can see that the bacteria or the harmful product has been internalized. And after the internalization, it is actually because of different type of extensions which are known as pseudopodial extensions from the leukocyte, and those leukocytes can either be neutrophils, monocytes, or macrophages. And this specific type of vacuole which is created is known as phagosome. Now this phagosome combines with another specific type of vacuole which is known as lysosome. This lysosome has different type of digestive components which when combined with phagosomes makes the phagolysosomes. And phagolysosome is actually the combination of both where the digestion or the killing of that bacteria or that product occurs. After that bacteria or that product is killed or degraded, it is then moved out of the cell as the debris. Now, in the phagocytosis, when anything is engulfed or internalized and it is degraded, the question arises is that how is it degraded? So it is degraded, or the microbe or the microorganism is killed via two different procedures, either the oxygen dependent or either the oxygen independent. The oxygen dependent microbial killing is the most important type of degradation phenomena, because in this oxygen is utilized and it is more effective. It occurs via the activity of hexose monophosphatase shunt, which supplies electron to NADPH oxidase in a phagosomal membrane. Phagosome is that vacuole in which the internalized microbe or the product was present. This results in the formation of different reactive entities such as superoxide ion, which is then converted into hydrogen peroxide by dismutation, which is then converted into hydroxyl ion. These hydroxyl ion can ionize different microbes or different proteins and render them or kill them. Myeloperoxidase halide system of bacterial killing is perhaps the most effective way to kill the bacteria or the microbes, in which hydrogen peroxide oxidizes the microbial proteins and disrupts the cell wall of the bacteria or the microorganism. In the presence of the myeloperoxidase enzyme and halide. That's why it is known as the myeloperoxidase halide system. Myeloperoxidase is actually an enzyme. On the other hand, halide are actually a reactive form of nonmetals, for example, chlorine, bromine, iodine, and fluorine, etc. And the most commonly applied here would be the chlorine. Now, the second process is the oxygen-independent microbial killing, and it is less effective as compared to the oxygen-dependent microbial killing. It is mediated by proteins such as lysozyme, lactoferrin, major basic protein of eosinophils, bactericidal permeability increasing proteins, and defensins. But whenever possible, this is the more suitable or more effective modes of degradation, which is the oxygen-dependent microbial killing. Now, what are the different mediators of inflammation? The mediators of inflammation can either be exogenous or endogenous. Exogenous, as the word shows, they are from outside the body. They are not produced in the body by humans or by our own tissues. So that's why the name exogenous. So exogenous mediators are actually released, or they come from the microbial origin, or for such as a bacteria. As you can see here, like formilated peptides of Escherichia coli, which is a type of a bacteria, which are chemotactic for neutrophils, and neutrophils are the first line respondents in the acute inflammation within 24 hours. The second is the endogenous mediators, which for which the source is our own body. So it can be the host origin, or the vasoactive amines, the arachidonic acid metabolites, cytokines, the kinin system, the complement system such as C5a and C3a, etc., and nitric oxide. Now, there are different types of conditions which can actually impair the process of inflammation. And as we can see in all those previous slides, the process of inflammation is very important for the integrity of a human being. So these hereditary defects are of different types. It can be either because of the deficiency of the complement systems such as C2, C3, and C5, which are actually the mediators of inflammation, or it can either be because of the defects in the neutrophils, such as chronic granulomatous disease of childhood, which is an X-linked disorder. So it is genetic and hereditary. In this process, the NADPH oxidase activity deficiency occurs, which results in inability of the phagocytic cells such as neutrophils, monocytes, and macrophages to kill the microorganisms, even though if they can ingest them. Another defect or hereditary defect is myeloperoxidase deficiency, which results in recurrent infections, but it is not of great consequence. Then there is Shediac-Higashi syndrome. In the Shediac-Higashi syndrome, it is actually an autosomal recessive syndrome, and it is observed in neutropenia, albinism, cranial and peripheral neuropathy, repeated infections, and abnormal leukocytes or white blood cells. And the last type is the leukocyte adhesion deficiency syndrome or disease. In this, there are recurrent bacterial infections, and as you can see here, that the problem is in the adhesion molecules. And which adhesion molecules are involved? There is type 1 and type 2. In type 1 leukocyte adhesion deficiency, the beta-2 integrin deficiency is there, and in type 2 leukocyte adhesion deficiency, there is mutation in the gene which codes for fucosyl transferase. So in this lecture, we discussed the cellular responses to leukocytes, mediators of inflammation, and hereditary defects which can impair the process of inflammation. But still, there is a lot more to learn, and so please stay tuned to sqadia.com and keep on watching. Thank you very much.